in the Democratic Republic of Congo on sickle cell anemia. And before I dive into this work that we are doing currently, I wanted to bring us back to where and when the research on sickle cell disease anemia started in DRC. We are the third biggest country in Africa with more than 90 million inhabitants. And in our country, sickle cell anemia account for 2% of the newborns. So we are, from a geographic point of view, well positioned to inform on migration, gene flow, and how could have influenced the adaptation and the population distribution, and also could have an effect on the phenotype and the response to treatment as well. The first phase of research on sickle cell anemia, as far as I was able to retrieve in the literature, the first publication was made in 1952 by a team of researchers from the University of Leuven. This is the mother university that actually was at the origin of the University of Kinshasa. And the work they did from this publication of 1952 to 1966, this is the last publication they put out. It was more about describing the phenotype and also trying to understand how other diseases could affect the phenotype of sickle cell diseases. And the second phase was led by the late Professor Kabakele. He was a surgeon and was specialized in bone diseases. He was a traumatologist. And he came to the field because sickle cell patients, they always have the wounds that are not healing, and those ulcers. He was one of those that developed surgical approaches on how to treat those patients. So he was also at the origin of the biggest sickle cell research center in our country. The third phase, I believe this is the phase we are in now, and one of the pioneers in this third phase is Leo Chilolo, that most of us know very well. He has been conducting researches since 1992, starting by describing the phenotype, and now he has dived into exploring also the genetics, which genetic factors in our setting would influence the phenotype of sickle cell anemia and could also influence the response to the treatment. Although he has not yet published this part of the study, that we know that he is already looking at this type of approaches as well. And then we, the Center for Human Genetics of the University of Kinshasa, we also came into this field very recently, and these are our few publications from the description of the clinical phenotype, the description of the severity of the phenotype in Congolese patients. We have also quickly evolved into studying the genes. This is our core business, looking at the genes and how the genetic determinant of one individual could affect his baseline phenotype. And this publication that we made in 2018 was actually the one that built or established the foundation of the work we are doing now. From the moment that we could replicate that genetic factors are modifying the phenotype in Congolese patients, and from the fact that more and more publications are being put out that are showing the influence of the same genetic factors on the response to hydroxyurea, and additional genetic factors that could also influence genetic response to this well-used treatment, we thought that it would be interesting to go further. In the first study, we explored 8 polymorphisms, and we thought that it was time to increase the number of polymorphisms that we could explore. So we developed another study which remains a candidate gene study, because we are not conducting GWAS, but we are targeting SNPs that has been published in the literature or deposited in GWAS catalog, where there is a strong association that has been established between the gene and either the baseline values of fetal hemoglobin or in association with an effect or the metabolization of the hydroxyurea and other SNPs that have been associated not with the baseline but only with the effect of hydroxyurea. So from our literature search, we actually identified 56 SNPs that we thought that would be worth studying in our population. This included 13 SNPs that had been previously associated to the baseline levels of fetal hemoglobin, 28 associated to the effect of hydroxyurea on fetal hemoglobin, and we also had 10 polymorphisms that have been associated with both the baseline value and the level after the treatment with hydroxyurea. We also included one SNP that has been associated with the liver metabolization of this drug. And in addition, we also wanted to look at the haplotypes of sickle cell anemia in our country, since this is one of the information that were not available yet. We commonly use the imputation from data that has been published in the neighboring countries. We were lacking data from our own population to know which haplotypes are common and how those are distributed. So we took this as an opportunity to go beyond and try to explore the haplotypes in our community. This study was conducted in 2 hospitals. One is the center that I talked about in a couple of slides ago, that was developed by Professor Kabakele, which is the reference center for sickle cell anemia in DR Congo. And the name in French is Centre de Médecine Mixte et d'Anémie SS. And this one is located in Kinshasa, the capital city of Congo. But the study was also conducted in a rural area in the hospital named Saint-Luc Hospital in the province of Congo Central. So we combine the urban and the rural setting for this study. One of the things that is important here in the rural setting, this particular hospital is only looking after pediatric patients. So we excluded pediatric patients from the urban setting so that we could have a sort of representation where we had children from one side and adults from the other side. All the information and samples were coming to the Center for Human Genetics of the University of Kinshasa, where we were able to actually perform hemoglobin typing using capillary electrophoresis and to extract DNA from those patients. We were also able to do the confirmation of the diagnosis through a molecular testing, and Solomon talked about, and the next speaker is also going to talk about. We developed this approach because many of the patients that are labeled as sickle cell anemia, and some of them are even receiving treatments, are actually not sickle cell anemia. They have other problems, but they are not sickle cell anemia. So to be included in this study, we had to conduct this test, to perform the molecular test to confirm that the patient that is included in this study is truly a sickle cell anemia patient. And then DNA was sent to the K11 in Belgium, where PhD students within this project are registered. This is where they are actually doing the PhD thesis. So to be included in this study, patients needed to be part of the cohort of sickle cell anemia in one of the institutions where we were recruiting. The second, we had to validate the sickle cell anemia status using a DNA-based test. It's a resection fragment length polymorphism test with the digestion enzyme that is actually available at the University of Kinshasa. The patient have to have not received a blood transfusion the 3 months preceding the recruitment. Of course, they have to provide a consent signed either by other patients or by parent when the patient was a child. They had also to be available to provide blood sample, not only for the recruitment, but also for the follow-up that were set in the frame of this study. So after the initial assessment, which included the recruitment, the consent process, and the formal recruitment, the primary clinical examination, and based on the primary values, patient was actually classified into 2 groups based on the level of the fetal hemoglobin. Those with fetal hemoglobin equal or above 15, they only received the molecular result of the DNA test we performed at the University of Kinshasa, but they could also keep coming to the 3-monthly consultation that our team set in either of those institutions. But for those who have the baseline level of fetal hemoglobin below 15%, those were included in the trial that we set up. So in this trial, the hydroxyurea was provided to patients starting with a dose of 10 to 15 milligram per kilogram. And we used an escalation of 5 milligram level, and this was a monthly escalation until we reached the maximum tolerated dose, which was 30 to 35 milligram per kilogram. Every 3 months, all the patients were seen at a consultation in the institution where they were recruited. During those visits, a clinical evaluation was performed. We were looking at the adverse drug reaction when they have not been reported within the window of 3 months. We actively looked at those adverse drug reactions. We also sampled for blood testing to control for fetal hemoglobin, white blood count, reticulocyte platelet, and LDH. So at the end of the trial, this is the operational definition we use to separate patients between responders and non-responders. This is a definition that we borrowed from this publication, where a respondent was someone who had reached a plateau of fetal hemoglobin above 20%, or someone, even if the plateau was not reached, but someone that tripled the initial level of the fetal hemoglobin. This one also was considered as a respondent. And this is the simplified description of the single molecular inversion probe library preparation, where you have this construct that is designed by the manufacturer based on the data that we provide, the target that we provide to the manufacturer. And this construct includes at either end a target that is specific for the sequences that are flanking your SNP of interest. And then you have a tag that is specific to that particular SNP. You have a primer site that will help to amplify this fragment. And when these fragments is hybridized on DNA, the sequence that are specific to the flanking of the polymorphism that you are exploring, this one is the one that is going to ligate actually to your native DNA. And this will circularize during a PCR process. Non-circularized DNA will be removed by exonuclease. So at the end, you have your fragment that has been amplified. They have been actually tagged with a patient-specific code bar and are multiplexed. This library is going on to the MySeq. This is where our sequencing was performed as a paired end on a flow cell with 200 cycles, 2 times 100 cycles. So what we have observed, this is the general characteristic of the population. Before we go into the clinical trial, our cohort consisted of 275 individuals. So the distribution of genders was actually skewed, as you would see on this graph of the haplotype that we are just showing this, not to discuss the haplotype, but to give you a visual idea of the gender distribution. As patients were older, we observed that there were significantly more female than male in our cohort. The age ranged from 1 to 40 years. At the baseline, at the beginning, when we had the 275, we looked at the baseline values. What we observed out of the 275 is that there was a sort of difference that was very significant for the fetal hemoglobin, the hemoglobin in general, the fetal hemoglobin, the white blood cells, and the reticulocytes and platelets. When we compared Kinshasa to Kisantu, as I said, Kinshasa were adult patients and Kisantu children. So we observed this difference between adults and children. But for some of those parameters, the significance was not confirmed when we performed the Bonferroni correction. For those who were included in the trial, in total 105 were initially included in the trial, but 25 were later excluded, including 2 that were deceased. They were excluded for diverse reasons, and this is not discussed in this presentation that we are giving today. The age ranged from 2 to 38 years. So before the hydroxyurea started, when we zoom into the initial data for the 77 patients that finished the clinical trial, we observed some differences in the value of the fetal hemoglobin between genders and between ages. This value was higher in female and was higher in children. After the clinical trial, when we separated patients into respondent and non-respondent, we observed more respondents than non-respondent in this cohort. And when we compared the proportion of respondents between children and adults, we didn't observe any significant difference for that. That cannot be ascribed to the age of the participant. So here we are presenting the evolution of the fetal hemoglobin over the course of the 24 months of the research. As you would see, we observed a significant increase in the fetal hemoglobin in the group of respondents. This is actually the main criteria that define respondent between and non-respondent. So in a way, this is a kind of circular reasoning because this is the only criteria that is separating the two groups. You can see that there is an evolution, there is an increase within the two groups, but this increase is much more higher in the group of respondents. But when we compared the respondent and non-respondent for the white blood cells, we did not observe any difference that was created for the white blood cells. In both groups, we observed that the hydroxyurea treatment actually decreased the amount of the number of white blood cells, but the decrease was not significantly different between the two groups. The same trend was also observed that we could see that there was a change in the value of reticulocyte, but the difference was not significant. The same also applied for the LDH. And when we look at the data that were generated by sequencing the 56 SNPs that were retrieved through the literature, the first thing that we did is to make sure that the variation that we could see looking at the results of the clinical trial would not reflect any change that would be at the population level. So the first thing that we did, we explored the Hardy-Weinberg equilibrium for each of those SNP at the level of the population, which is the 275. So out of those 56 polymorphisms, 4 were actually not balanced in the population, which means that there was a skewing. They were not fitting with the expected frequencies for those SNPs in the population. So those 4 SNPs were further excluded for the clinical trial evaluation. When we evaluated the remaining 52 SNPs with regard to the response to the hydroxyurea between the respondent and the non-respondent, we did not observe any significant influence of for 48 SNPs, only 4 actually showed a significant correlation or influence on the level of response achieved, or the level of fetal hemoglobin that was achieved at the end of the clinical trial. So we have actually, as I have shown through the story of sickle cell anemia in DR Congo, set an environment for genomic research on sickle cell anemia in DRC. And this study illustrates the complexity and the difficulty of running a proper clinical trial in a research-limited setting. We have observed that some of the well-known trends were also present, but the significance was not reached. We don't know whether the sample size could be one of the reasons, but only 4 SNP actually correlated with the level of fetal hemoglobin expected after hydroxyurea treatment. Even some of the correlation we observed in our previous study of 8 SNPs were not significantly confirmed here. So maybe a GWAS is needed to explore Congolese patients to identify much more SNPs that could be associated with both the baseline phenotype and the response to hydroxyurea. We hope that with further collaboration and fundings, we would be able to dive deep into the genotype influence of the baseline phenotype and the hydroxyurea treatment in Congolese patients. So I would like to thank the funders. This research was mainly funded by the Rios, and we also received a strong support from the University of Leuven in Belgium. And this is the team that is driving this research. Thank you very much.